Hello and welcome to another biology lesson. We are once again going to be looking at cladograms. Today we're going to look at some of the challenges that actually come up in making these things. How do we decide how to group things and how we overcome those challenges? And so you know that hopefully that these are made based on derived features and so or derived characteristics. And so the idea is the more derived characteristics two organisms share, the more likely they are to be closely related. But there are some challenges, and we've already kind of mentioned at least one challenge, and that's with vestigial structures. And so we talked right here, we have this branch of sharks, and then there's this derived structure here, the four limbs. And so this all becomes clad tetrapoda. I couldn't remember that the last time we talked, but I remember now. Tetrapoda is that clad, that means four limbs. And so everything here should have four limbs, except that when we get to the snake, it has no limbs. When we look at a whale, arguably two limbs, or again, none, depending on whether or not you would consider the front fins a limb or not. And so we have this issue that features can be lost. And so how do we cope with that? Obviously, one way to look into that is to say, let's see if there's any evidence in the fossil record or in the living organisms of vestigial structures. Are there vestigial bones? Some snakes do have vestigial bones from their limbs. Some whales might, certainly in the fossil record they do. So that's one way to deal with that. But another wrinkle that comes up are those analogous structures. Now, analogous structures, remember, they're used for the same purpose, and so they're often very similar, but they have a different structure. So they derived from a different original structure, and so we can look at, well, sharks have fins, and so do whales, but they weren't derived from the same original structure. The whale, actually, its fins were actually derived from these limbs that the shark didn't have, because the shark branched off before limbs were evolved. Similarly, we look at the bat and the bird, and there are some similarities in structure because they both did have this four limbs, so both their wings are derived from this original ancestor's four limbs. But the bird's was derived from some kind of reptilian limb, whereas the bat would have been derived from some kind of mammalian limb. And there would have been, by the time that these wings developed, there would have been structural differences between the limbs of a mammal and the limbs of that reptile. And so those structural differences carry over to now. And so there's also some structural differences. But the wings look, by appearance, very similar because they're used for the same function. And so it'd be very tempting to say, oh, wings is a derived feature. And it could lead us astray on that. And so the way we look at that, or with modern technology, one of the ways we can cope with that is by looking at the genes. And the one set of genes that is especially useful is the DNA found in mitochondria. So you may recall that I have mentioned previously that mitochondria has its own DNA. I think I mentioned that, maybe I didn't. If I didn't, hey, mitochondria has its own DNA. Mitochondria reproduce by themselves, kind of like cells do. They duplicate their DNA and multiply within the cell. Of course, they are the powerhouse of the cell. They make ATP. They're incredibly important. And the thing that makes them really useful in cladograms is that all eukaryotic cells have mitochondria, which mean all multicellular organisms, whether it's a tree or a shark or a whale or a person, we all have mitochondria. We all have mitochondria DNA. And there's not a lot of DNA there. And so it's something we can kind of analyze. And since every organism needs ATP, a lot of the proteins that are created by that DNA in the mitochondria is very similar because all those proteins are trying to do the same thing, which is make ATP. And so we can look at how different that DNA is, how different those proteins are. The more different they are, the more distance we think there are between common ancestors. And the more similar that DNA is, the more similar those proteins are, the more confident we can be that those two organisms shared a recent common ancestor. All right, sorry. So if we look at this, if we look at this, we can see I have a couple different birds here. We have the hooded vulture, the American vulture, and the stork. Now, if we were just looking at those pictures and based on the physical characteristics, we would look at that and say, clearly, the two vultures look far more similar. They seem to appear to have more similar characteristics than the stork does with either one of them. But if we analyze their DNA, and I made up this chart, so I have no idea what the real number of differences between their DNA is. But let's say we look at their mitochondrial DNA and we see something like this. So I'll explain this a little bit. What I've got each animal across the top and each across the side. And so stork here, we're going to compare how many differences are there in the DNA between the stork and the stork. Well, they're both storks. There ought to be none, right? They have the same DNA. So then I compare the stork to the American vulture, and I see there are 4 differences in their DNA, and the stork to the hooded vulture, and then there's 8 differences. Now we'd be looking at one specific protein or one specific gene when we're doing these comparisons. It wouldn't necessarily be the entire genetic code. Though I suppose with technology maybe we could do the entire genetic code, but usually what we're trying to find is a section of DNA that we think codes for basically the same thing. At any rate, then I can come down here and compare the American vulture. Now, American vulture has already been compared to the stork right there, so actually I can go ahead and leave that blank. It should have no differences with itself. And then hooded vulture, it's going to have 9 differences. Again, I made that up. Hooded vulture, we skip these. So basically anything under this 0 line, we just leave blank because it would be repeat information. I could put hooded vulture compared to stork, but I have hooded vulture compared to stork right there. And so just to make it a little less, less data to look at, we leave half of it blank. Anyway, what you can see from this is that the stork must be more closely related to the American vulture than the hooded vulture. And the American vulture is also more closely related to the stork than the hooded vulture. So you can see with 9 differences here, but only 4 there, the American vulture is more closely related to the stork than the hooded vulture. And so that would be maybe something that would be surprising and we wouldn't expect and maybe wouldn't even be able to figure out without this DNA analysis. So if we were to actually draw a cladogram for that, it might look like this. We'd have some kind of ancestral bird. It branches and we get the stork and the vulture, and then it goes to the hooded vulture over there with the common ancestor further back. So these two are more closely related. They share a more recent common ancestor, and this is less recent than that. And so since these two had about the same, one was 8 and one was 9, they had about the same difference from the hooded vulture. I wanted to put them the same distance from the hooded vulture. That's why they both branch off together. Hopefully that makes sense. So if you go back and pop it back up real quick, both the stork and the American vulture were about 8 and 9, about the same number of differences. So you would think that they would have about equally related to the hooded vulture, and so they split off together and then separate. All right, I hope that makes sense. DNA, using DNA to analyze the relationships has made a huge difference. It's changed a lot about how organisms are organized into clades, and it's changed where we put things, because, like I said, the way they looked and the way what DNA tells us don't always line up. Anyway, thank you for watching. Take care, and yeah, that's it.